Hello, everybody. Welcome to the BINA AIMM user group. So I want to, being located in Canada, I want to first start by saying that I respectfully recognize and acknowledge the relationship that the First Nation, Inuit, and Métis across Canada have with the land that all Canadians live on and enjoy. I recognize human rights of people who have suffered from systemic racism and discrimination in Canada. I seek to provide a welcoming, engaging, and safe community for everyone. Thank you. All right, so again, welcome to our AIMM user group. I'm Judith, and I co-chair this group with Katrina Strambio de Castilla. Today we're going to have a nice and I think overdue presentation about ROR and RRID and PIDs for organizing metadata into the imaging field. First, we're going to have a presentation by Amanda French on the research organization registry and how this connects with bioimaging facility. Then we're going to pursue with Nate Herzog, who's going to talk about the PIDs in systems to organize facility data management. And we're going to end the presentation with Anita Brandowski, who's going to talk about RRIDs, what they are, why do they exist, and what do they cover and not cover. So we're going to start with Amanda. Amanda, welcome to our user group, and we look forward to hear your presentation.
Thank you so much for having me. Let me just share my screen here very quickly. Oops, make sure I'm sharing the correct screen. Share. Here we are. Okay, so you should be able to see my presentation now. And I do apologize, I changed titles on you a little bit. My original title was The Importance of ROR for Facilities and Instruments. And I realized that my talk is really a bit more about the unimportance of ROR for facilities and instruments. Once we get through the whole presentation, you'll see a little bit about what I mean. So this is a bit more general about, what do you need to know about ROR? When is it appropriate? When is it not appropriate? And then Anita and Nate will tell you a lot more about RRIDs later in the presentation. So first, I'm going to give you a brief introduction to what ROR is, and then we'll talk specifically about the facilities and instruments use case for ROR, or as I mentioned, lack of use case in some cases. So what is ROR? Why is it important? ROR is a global community-led registry of open, persistent identifiers for research organizations. You all may or may not be familiar with the term persistent identifier, but essentially it's like a social security number. It helps disambiguate entities from one another when they may have the same name or variant names, that kind of thing. We're established, trusted, widely adopted in the scholarly communication landscape, and entirely free thanks to the support of our operating organizations, Crossref, DataCite, and the California Digital Library. I can talk a bit more about our sustainability model if you like, but I thought that might be a little out of scope. Probably most of you have heard of DOIs and understand what DOIs are. I work for Crossref, which is one of the organizations that creates DOIs, not the only one. DataCite is another organization that provides DOIs, and typically, although this is a very permeable boundary, typically Crossref registers DOIs for published content like journal articles, whereas DataCite registers DOIs for data sets. There's lots of overlap, and any of them can create DOIs for anything, but that's sort of typically how it works. And as I mentioned, those are two of the operating organizations of ROR. So DOIs are identifiers for research outputs, including journal articles and data sets. ORCID is a persistent identifier for people. We need to be able to tell people from one another just in the same way, and on a time scale of hundreds of years, which is the time scale of scientific research. So that's where the persistent comes from. And then you'll notice the founding dates of each of these organizations. 1997, I mean, first, the most important thing was to get links to research outputs that were not going to go away, that would be persistent. The next thing was to try to tell people apart from one another. And then most recently, ROR has become the open persistent identifier of choice for most research organizations. Oh, and of course, I mentioned as well RRID on this, which is another key open identifier, which Anita and Nate will tell you about. So here is an example of a ROR record. We have an openly searchable, essentially, catalog on the web. I encourage you to go to roar.org/search even while I'm talking and look around. But maybe before you do that, let me go over the elements in a ROR record, at least the ones that are shown here. The ROR ID itself is here in the upper left. That's an automatically generated opaque identifier that always follows the same pattern. You can't choose a ROR ID. We assign them centrally at ROR. And we prefer that people use the ROR ID as the URL itself, because it means you will, the link to this page is that ROR ID. And having identifiers as URLs means you can always resolve them. Even if we were to move to another organization or something, which I have no intention of doing, we could always point those URLs at something else. So it's important to be able to see what that identifier is representing. So you can see the name of this organization. You can see the types of what type of organization it is. This organization, many of you might be familiar with it, is an arm of NIH that is itself a facility. It runs labs and things like that, but primarily it's a funder. So it's both a facility and a funder. We have other names, acronyms, especially in other countries besides the US, we'll typically have a lot of names in other languages. So in non-English speaking countries, there's almost always an English name and then a name in the language of the country where the organization is located. ROR has global coverage. We take that very seriously. We try not to be any more Western biased than we need to be. You can see in the bottom left here, what organizations is this organization related to? This is a sort of a sub-organization, one of many of NIH. Where is it located? What is its website? And what are some other identifiers that identify this organization? We currently have over 116,000 research organizations from around the world. And even though you can see this nice record on the web, also in that bottom left, you can see the JSON metadata, which is really the key part of the ROR ID. The key function of the ROR ID is to associate that ID with information about that organization that can be used by software systems. So you can click to see the full record metadata, which includes a little bit extra metadata. And if you think there's something wrong with this record, you can go right to this record and suggest a change via that link in the bottom left. Here's the problem that ROR was founded to solve. Just as people may go by different names, they may change their name, they may change their, they have a maiden name, a married name, or all kinds of reasons. But even similarly, organizations usually go by multiple names, either in multiple languages or just nicknames. I used to work at Virginia Tech, which is why I use this example. This is a search system. These are real screenshots from a search system that shall remain nameless. But if you search for Virginia Tech, you get different results than if you search for Virginia Polytechnic. The real name of the organization, to be honest, I can never remember it because everybody calls it Virginia Tech. But the real name of the organization is Virginia Polytechnic State Institute or something like that. So we can see as humans that these are the same organization, but this system can't tell that at all. And that's a problem. So what you need to do is have identifiers, unique identifiers, where it's not just text strings that could be multiple versions, but here's an identifier for this organization that is associated with multiple name variants in particular. So ROR solves this problem. ROR is the majority of organizations in ORCID. ORCID has been a tremendous ROR adopter. They are also using RRIDs, but most organizations, so particularly for what organization do you work with, what organizations have you received funding from, all of that kind of thing are usually identified by ROR IDs in ORCID. You can see this in your own ORCID record. If you go to your profile and click on show details, you'll see which identifier is identifying your organization. And so ever since 2019, more and more ROR IDs have been getting into ORCID, and now it's a vast majority, and I think in a year or two, almost everything in or almost all organizations in ORCID will have ROR IDs. Just some general features, over 115,000 records, totally free to use, thanks to the support of Cross-Reference DataCite and CDL. Really designed to be integrated into software systems, which I'll talk a little bit more about in a minute. Highly multilingual metadata, all the character sets, Hebrew, Cyrillic, various Asian data sets, all of that. Organization hierarchies and relationships. We try to do our best to be quick when people want to add a new organization to ROR or change the information in a ROR record. Usually 4 to 6 weeks is our processing time for that right now. Mapping to other identifiers. And then we offer a lot of sort of tools and services to where, like, if you have a spreadsheet with a bunch of names, you want to match that to ROR IDs, we have tools that can help you with that. Lots of systems are using ROR, Crossref DOIs, DataCite DOIs, ORCID, as I mentioned, search indexes, including Web of Science, OpenAlex, Lens, lots of repository and CRIS systems. So a lot of the repositories where you're going to upload data sets will have ROR, whether you see it or not. And then publishers are a bit more slow, but a lot of publishers, actually Optica Publishing Group, which I imagine some of your researchers publish in a lot, is a tremendous ROR user. They're really fantastic. But lots of publishers are beginning to use ROR more and more. So let's talk about ROR for facilities and instruments. When is ROR used for facilities and instruments? As we saw on the previous record, we do have an organization type of facility, which we define as a specialized facility, maybe a bit tautological there, where research takes place, such as a laboratory or telescope or dedicated research area. Now, the key to the difference between ROR and RRID in particular is that we are an organization registry. So we create identifiers for organizations. So some facilities and even some, I want to say instruments, but it's not really the instrument, it's the organization that manages the instrument. When those are mostly separate or well-defined entities, organizational entities, or, and this is kind of a key thing, when it's an organization that is supported by multiple organizations. And really particularly when it's an organization that is supported by the federal government. Those tend to be the facilities that are in scope for ROR. I was looking up, so I actually, in one of your introductory slides, you have an upcoming speaker who's associated with the facility. That facility is based at a university. Most likely that, ROR has universities, out the wazoo. We have all the universities, not always the research facilities, because so many of them are dependent, they're sub-units of an organization. So I'll just give you some examples here. Here are some international bioimaging facilities in ROR. I sort of spot checked the members of this group. Most of the core facilities who are members of this group are not in ROR. And again, we'll talk a little bit more about this later. But we did see a national research facility that is in ROR. And in other countries, especially in Europe and Asia, some of these institutes are supported by the federal government, much more so than some of the core facilities in the US are, or they're supported by multiple universities. France in particular has a lot of these types of organizations. So there are a lot of facilities in ROR, but they tend to be those separate or nationally funded organizations. And like so here's all these centers for microscopy, but like a particular microscope will not be in ROR. There's not an instrument really for ROR. And not all centers for microscopy will be in ROR if they're only a subunit of an organization. So hopefully that's a little clear. How do ROR IDs and RR differ? Anita and I collaborated on a blog post. We get a lot of hits on this blog post. This is very popular on the ROR website. So I encourage you to read this blog post. I will even put the link in the chat here. And I was surprised actually when we did this to see how little overlap there is between the entities in RRID and ROR. I thought there would be more, to be honest. Facilities and instruments in ROR are organizations. They may be associated with a university, usually multiple universities, but they're not dependent on it. Core facilities, which are identified by RRIDs, are usually not in scope for ROR because they're subsections of a university or they're dependent on a single grant or both. So ROR IDs, I mean, like there are some rare cases where you can use a ROR ID to identify a facility. We have facilities in ROR, but the more common use case for ROR IDs is to identify contributor affiliations. What organization is this person employed by? Or funders, and we'll talk about a little bit about this. And this is where those edge cases get weird, because sometimes facilities are considered funders because you're providing in-kind funding for a researcher, right? And two, for some research projects, the staff who is affiliated with a particular facility may have contributed so much to the research that they're listed as an author on or a contributor to a data set or an author on the research, right? So there's all these little weird edge cases, but in general, ROR IDs are used to identify contributor affiliations and funders, even though we have a facility type, etc. And then the other thing that is kind of quite different between ROR IDs and RR IDs, and again, these are generalities, but in general, RR IDs tend to be added to text of a manuscript or data set. They're in the description, they're in the funding acknowledgements, they're like, hey, thank you to the support of such and such facility for use of your facility. Whereas ROR IDs were designed from the beginning to be built into software systems. So that's typically how ROR IDs are used. And what that means too is that researchers do not need to know ROR IDs. We almost discourage it. Please don't tell your researcher what their ROR IDs are. Even those of you who are staffing facilities probably don't need to know your ROR ID. It's designed for research workflows. And I'm going to show you sort of a real example of this and then wrap up. Oh, this is breaking news. This doesn't really fit in this presentation, but this just came out yesterday, so I thought I would mention this. Many of you may be familiar with bioRxiv, which is for preprints, not data sets. But this is one of the systems where ROR is built in invisibly, and you can kind of see down on this announcement. Here's this link to this announcement that they've just implemented ROR, that they're using ROR for a funder pick list. And I'm going to show you how that works. I just thought this was exciting and then just came out yesterday. So here's how this works. So this is a data set repository, a data repository called Dryad. Some of you may be familiar with this. You cannot see a ROR ID anywhere on this screen, but it's ROR that is powering these lookups. So these form fields could be text fields. They could be just type in the research facility where you worked, type in the funding organization, and if that's the way this system worked, then the data would be all messy. So ROR helps clean up this data. Dryad uses ROR for this research facility lookup, which means that it probably won't have a lot of core facilities in it, but it does allow you to type the text if it's not in ROR. They might want to build RRID into this at some point, I don't know. But the granting organization, you can see, is also powered by ROR, and that tends to be almost all the funding organizations you can think of are gonna show up in that pick list for ROR. So this granting organization thing is exactly what bioRxiv has just implemented for its preprint server. Can't see ROR except in the logo on the bottom right of the slide, but this is ROR in action. One of the things I kind of liked about this Dryad is that whether or not they should be using ROR for this research facility lookup, it's great that they have that field because most systems don't, right? This is one of the problems, is that in published research articles, in data sets, there's not a place in the form for people to say, this is the research facility where I did this work. So I just think that this is my opinion. This is what the problem is that more of these platforms, and there are tons of them, publishing platforms, data set platforms, all of those should at least have somewhere where you can say, here's what the research facility is, and it should be in a structured field. That's really all. We can go to questions now or save them till the end.
All right. Thank you very much. Look, for the sake of time, I think we're gonna move on to the next speaker, but do please either put your questions in the chat or keep them for the end, and we'll come back to questions at the end, because I know that there's already quite a number of them.
Yeah.
Oh,
So
thank you.
Maybe Amanda, you can check.
Yep,
Yes.
I'll go answer questions in the chat.
Yes, excellent. All right, thank you. So we're going to move on to our next speaker, Nate Herzog from Vermont Biomedical Research Network is going to talk about PIDs in systems to organize facility data. Nate, the floor is yours. You're still muted.
There we go. Okay, hi everyone. As Judith said, I'm Nate Herzog. I'm out of the University of Vermont in Burlington, and I'm the project lead of the core marketplace. I'm going to be giving a talk mostly not about what PIDs are, but then how we use them, sort of piggybacking on kind of the end of Amanda's presentation. So there we go. The core marketplace, if you've never heard of it, you can find it at coremarketplace.org.
Would it be possible for you to go in presentation mode, please?
Am I not in presentation mode?
No.
It's not showing up
on screen.
Oh, interesting. It is on mine. Okay, hold on. Oh, it's being paused. Why is it being paused? How do I not pause it?
I mean, if it's too difficult, it's okay.
No,
I can share
Hold
this
on.
this slides for you if you
hold
want.
on. No, no.
All right. Holding.
Let's see here. Tell you what, can we do it this way? It's a little ugly, but it works.
Now we are seeing your email.
My email. You don't want to see my email. Good gravy. Where is it? Where did it go? There. Yes?
Yes, now we're seeing it.
Yes.
Okay, we're seeing
Excellent.
it's not in presentation mode, but it's fine.
It's fine. You can see it. Okay, I don't know. In the interest of actually presenting data as opposed to getting my computer to work, let's just stick with this. Okay. The core marketplace, as I mentioned, is an international repository of core facility information. It lists a number of different major data points like core specialties, instrumentation, publications that they've mentioned in their institution, their location, contact information. It mentions related facilities, both within and without their institution. And the core marketplace is a system that leverages PIDs heavily. We use RORs for the institutional data. We use ORCIDs for the the login information for various persons, so they don't have to remember passwords. We just borrow the ORCID system. We use RRIDs to identify both our facilities and our instrumentation. So, and the purpose for all this, again, is just to help facilities organize their data a little better as much as we can. To learn more, if you want to, go to coremarketplace.org. I'm not going to spend a whole lot of time showing off the individual features of the site because I really kind of want to dig into the problem that we're trying to solve here with PIDs. So I think it doesn't take a whole lot of arguing when I say that facilities create a lot of data. everything that comes out of their instruments to everything that goes into the publication at the end and everything in between. There's a ton of data. And because facilities are run by humans, anybody who lives in a house with a basement will know that if you have things that are underfoot and you don't want to get rid of them, but you don't want to trip over them either, what do you do with them? You throw them in the basement. And when you throw things in the basement, you're not always careful about, how you're throwing them or organizing them necessarily. They can be badly labeled. They can be stuffed in bad boxes. They may not be stuffed in boxes at all. They might just be thrown into the corner. And facilities oftentimes will have what I call meta basements, right? There's just all kinds of places where you can stuff data. You can put it on digital drives, both on servers and on personal computers. There's physical spaces within the within the facilities that get this. And there's also internet spaces. There's web spaces galore, both within your institution and outside of your institution, that can gather your data for you. And these meta basements, the key feature with all of these things is that data can be easily stored, but not so easily remembered and retrieved, right? And so it's really hard to keep on top of everything. This is a common problem with every facility ever. And what we at the core marketplace, and I think a lot of us presenting today, we use PIDs as the primary instrument for discovering and accessing that data. There are a lot of different identifiers and they have a lot of different purposes. There's RORs, there's RRIDs, there's DOIs, there's ARCs. I forgot about adding ORCIDs, but they're also a very key identifier in the research world. And at this point, I would be remiss not to point out another project that I'm on, which is the FAIR Facilities and Instruments project, run out of Boulder, Colorado. You can see the NSF grant information right here in the middle of the slide. But the primary purpose for this project is to try and get to the real nitpicky question of, which PIDs should we be using for what, for when, under what circumstances? What do all of them do? Because, everybody has questions, basically surrounding which PID should I use? What should be labeled with what? How can I access it again? And these are questions that come up again and again and again in the most unusual of circumstances within this group. So let it be known that we are currently wrestling with this. And we are on year 3 of 3 with this project. So we will be producing results shortly. But I'm not going to talk much more about that. But when you talk about systems like the core marketplace, when you have PIDs in place that can automatically discover and share data, I mean, you can kind of think of it like an Amazon warehouse, right? Like if you had a whole bunch of stuff in an Amazon warehouse and none of it was indexed with any sort of identifier or organizational number, there's no way they would be able to find anything. Like it would just grind to a halt. But that's not the way they work at all. In fact, even your local Target store or whichever store has highly sophisticated inventory systems where everything is labeled and cross-labeled, and they know exactly what's in the store and where they can get more and how much is in the back room and all of that. And the same kind of situation is in play here with PIDs and scientific data. So if there is a PID in place, it is possible that you can discover and share data. We do that with the core marketplace and publications. We use the RRID, and when you actively cite the RRID in a publication and that publication becomes digital, then we can go and find it again quite easily using systems like PubMed or PubMed Central. And to date, we have 3,106 as of yesterday, I just counted, publications that we have linked to core facilities. So the problem of core facilities not being able to find which publications they've been mentioned in, we have addressed them, not solved them, but addressed them certainly using specifically the RRID assigned to a core facility and using that PID in publication citations so that we can go and find them again. Also, you can use PIDs for discovery. It's a slightly different model, but we use RRIDs to inventory the instruments that are within a core facility. So it is possible that you can go to the core marketplace and say, I want to find all of the facilities that use the same instrument that I have in my facility. And you would be able to pull that up quite easily because of the use of the PID. And because this is Bina, I'm going to steal Katerina's thunder a little bit with permission, hopefully, because it's really her project. But there's more of a, we're just launching a new imaging project. It is called the Frameworks Imaging PhD, Empowering Data Research and Reproducibility through Microscopy Communicated Defined Persistent Identifier Descriptions. 4-year NSF grant funded project run out of UMass Amherst. Katerina's the PI. From here on out, I'm going to state with a very large caveat that this is a brand new project. We've barely started work on it. And so every detail and every name mentioned from here on out is almost certain to change. So don't hold me to it. But we're going to talk about this in very, very broad strokes so you can kind of see like a new kind of system that we can build that can leverage PIDs to help facilities track and organize their data. So it's built on the Micrometa app. I'm sure many of you are already familiar with this. It's been in existence for a little while now. It is a software that gathers the configuration of information of imaging instrumentation, as well as modules, accessories, settings, and so forth, and gathers them together in a configuration document so that you can, track from day to day the configuration of the equipment on your bench as you run experiments. That already exists, but we're building on top of that now. We're creating, it was in the grant at one point, called the Micrometa Platform. Again, name will probably change. But what it is, it's a cloud-based application that facilitates the assigning of PIDs to all of the elements gathered up within the Micrometa app. So essentially, everything from the instrumentation to the components to the configuration files, everything gets a PID. Everything gets registered, and then that information is transferred back up into the cloud app so that it can be accessed by other systems. And then that cloud app can then share it out via publications, via queries, and via direct publishing to other systems, the 3 I have mentioned here, but also more. And one of the things that this does is, you have within a facility that is labeled with an RRID, you can direct publish not only the instrumentation that's within your facility, but the configuration and all of the components of that particular imaging instrument straight up to the facility profile on the core marketplace. And so we would know not only that a particular say microscope would exist, but also how it was configured, and we would have the exact configuration document that you could just download and reference yourself. And what that provides is a possible discovery pattern that was very, very difficult before, and hopefully it becomes a lot more streamlined as a result of the projects like this, and hopefully more projects that come after it. But you would have a possible pattern where a researcher would go, read an article that was published. That publication, that article would cite, the PID for not only the facility, but for the instruments used and the possible configurations of those instruments. That researcher then could find the facility profile on the core marketplace and, matching up the PIDs they read in the article, they could find the matching instrumentation and the configuration files. And from there, if they wanted to reproduce that experiment or if they wanted to push it further, they could download those configuration profiles and basically, assuming they have access to the same equipment, reassemble the exact same setup on their bench and then go forward with their work. The takeaways from this is that, with PIDs in place to kind of organize and index the various components within a research facility. The facility contribution is obvious, first of all, because it's stated quite clearly in the publication kind of profile of things, and also that research can be more easily reproduced. Because there's no question as to what components were used. They're all clearly labeled. Yep, this is my last slide. And this kind of opens the door to so many more possibilities, not only within imaging, but we can take the same concept and maybe even portions of this work and transfer it over to other instrumentation within cores, both bioimaging cores, and maybe even beyond that as well. And all this to say is that we're circling back to my first metaphor, is that it's all kind of cleaning up the basement a little bit. To be fair, data is not unique to the facility. It kind of breaks the metaphor a little bit. But the data that's generated by the facility needs to be discoverable and linked whenever possible, not only for the benefit of the facility, but also for the benefit of the larger research community. Because, I think everybody who kind of works in the field has got a stake in the kind of data and the quality of data that is produced by any given core facility. That said, there's always more to be done, and also this, hopefully this spawns more projects collecting, identifying, and sharing facility data. And then here's my contact information. Anita's coming next, and then of course Katerina, who is the lead on the project that I briefly glossed over. And I think the pattern is that we'll take questions at the end, so I will turn this over to Anita.
Thank you very much, Nate. So yes, so our next speaker is Anita, who's as you now know, is a collaborator on this project with Katerina. So Anita now is going to talk more about the RRIDs. And again, we're clarifying the context in which we use ROR versus RID. And Anita is going to dwell further on this. So if you do have questions for Nate and Amanda, please go ahead, put them in the chat. Maybe they can answer them right away. But for now, we're going to pursue with Anita and certainly come back at the end for more questions. Anita, it's all yours now.
All right, thank you. And I think Nate's got the slides up, so good enough. All right, so I'm going to be asking a question. Why do RIDs exist? What do they actually do and what do they not do, right? Which I think you've heard already several of these kinds of questions. So I will be focusing more on the RIDs, but I'll try to point out where they actually play nicely with the other identifier systems. Next slide, please. So here I am shamelessly stealing slides and images from Nate. So apologies for that, Nate, but yeah, I thought this was a very powerful slide. So identifiers do all serve different purposes, and they are actually best when they are assembled together into large ensembles of identifiers so that multiple things can be identified uniquely, not just one. Next slide, please. So I'm going to tell you about that one of those little ones at the bottom there, which is the RID. So this is actually a publisher-driven specification for identifying something called a research resource. And so these were created to address the need to cite individual research resources and also something that may be of interest to this group, reagents, especially things like antibodies, within scientific papers. So the scope of this is not necessarily filling in boxes. It is, in fact, for the investigator to go in, search for the things that they used for their particular experiment. And some of those things are, turns out, the facilities and the instruments that they have used. but also things like software, databases, antibodies, cell lines, tissues, organisms, and plasmids. All of those have RIDs that are governed by various community authorities for those things. These, again, work very well with in-journal workflows. So there are some constraints on these identifiers because they are intended to work with in-journal workflows. And they are, in fact, part of the XML metadata schema called JATS from version 1.2 on. So these can become metadata for journal articles. There is tooling currently to both add these RIDs to journal articles. And as I think Amanda mentioned, publishers are not always the fastest people to actually take on standards or pick up things that they that really could solve problems. But these things are there and they are being implemented. I'll point some of those out. But there is also tooling, and some of it actually Nate mentioned because it's extraordinarily simple to extract this key information from papers. I also want to highlight the fact that the database of RIDs, and in fact, this is combined out of many different databases, all of them are human curated records. So that also places some constraints in terms of what kinds of identifiers can be used for what kinds of things. Okay, and I mentioned the coverage. So I thought it would be interesting to take a look, next slide please, at when core facilities and instruments actually came into the RID project. So the RID project started actually in 2014. We had the registry of key resources a little bit before that, and we were starting to add that registry really came into existence in 2006, 2007, but wasn't really used for anything other than our project. So what happened here in 2013 is we actually worked with a group of researchers called under the Eagle Eye funding umbrella, and they organized a whole bunch of the cores. We got those records. We also worked with Science Exchange, whether that, I mean, it turned out to be relatively contentious as a project, as a company. But what happened there is a lot of cores did get registered with their RIDs. There were over 1,000 cores at that time, and if you can see those little gray bars in 2013 and 2014, those are the core facilities that we know are no longer in service. So in fact, it wasn't 500 and however many here, it was also an additional about 100 that were registered in 2013, but those have now been noted that they no longer exist or in any way that we can find them. A couple of instruments were registered at this early stage, but really there weren't very many at all. A few years later, in 2017, we had the National Institutes of Digestive and Kidney Disorders actually come to us and start to register, again, a lot of the diabetes-related cores. And you can see that, that the year after, very few cores were actually registered. And then something magical happened. So Nate and I and a group called Used It started working together. And what this group did is it actually brought something to these persistent identifiers. So RIDs were there, they were being used for other things primarily. But Nate has, he's a little bit modest, but actually the largest organization of biomedical cores, the ABRF group, is the backing, right? And it's the place where the core marketplace started. This is an organization of active core facility owners. I'm sure many of you know this organization. Some of you are probably members. And what happened here is that, there is a constant outreach to the core facilities. There is a consistency in these meetings and other things. And when Nate and I and others from the Used It group started working together, what you can see first and foremost is that, yes, there were some cores registered, but you can see also that every single year there are cores registered. And it's, even in 2025, which is only a very partial year at this point, and this was pulled about a month ago, we're already at the same level of cores as we had in 2024, and the year is very much not over. So this is an activity that is now growing. We are continuing to get more cores over time. And the same seems to be happening with instruments. So Used It, this group took on upon itself the registration of a lot of instruments, and there were a lot of instruments that were registered by this group. This was primarily within the Florida State University ordering system for capital equipment. They actually listed all of those with us. And then what happened is individual investigators and core facility owners started to list their equipment along with this. So again, this is all being driven out of Nate's project, out of the core marketplace, and out of the cores themselves, who are now coming in to register a lot of these instruments and cores. Next slide, please. So just to, not leave out what the pages look like, this is one of the cores that has been listed. And what you can see there is there's structured curated metadata. If you hit one of those IDs, the JSON ID, you can see, and actually there's also an XML version that's available of all of this nicely structured metadata. There are structured relationships. And so what you see here, hopefully, is that there's a relationship between this particular core and a whole bunch of instruments. And of course, there is the listed by ABRF core marketplace. And then the primary thing that people do come here for are the list of citations. So that's down at the bottom there, and I'll delve into that in a little bit. But next slide, please. So where do the other identifiers actually sit? So what's not explicitly listed on this page, although it is, if you keep going down on the to the bottom, there is the ROR facility, or the ROR university name for Stanford, and that is Stanford University. It is also, this is a curated page, and so we always put the university in front of the name of the core facility, because you can imagine that there's a mass spec core in every single university. There's an imaging core in every single university, sometimes multiple. So we at least put the university name there, and then that university name is also the parent organization of this facility, and that does have a ROR. When you log in to actually claim ownership of a facility, which we strongly encourage, that can be linked to the ORCID identifier. And of course, we even have now just implemented a nice two-way interaction with the core marketplace, where we are sharing a lot of this data. So with the goal of never having to do this data entry twice. Now it's a lofty goal, I understand, but we are working towards that. So we're really hoping that there will be additional interactions, especially through the recent funding, so that we can have the goal of never having to enter this stuff twice. Okay, so if you go to the next slide. Okay, so we have all this great metadata, and some of the great metadata, like multiple identifiers, some of those are coming from various places, including DataCite or other places. There might be a set of things that we can actually search for. So in this listing of this particular fluorescence microscope, And this is, again, the base model microscope that is present in multiple cores. And in this particular case, one thing that I'd like to draw your attention to for this particular instrument, this instrument is no longer in service. This is no longer, you cannot buy this anymore from the manufacturer. So one of the things that we have been doing is we've been collecting the specifications for these kind of instruments, especially the ones that are no longer available from the manufacturers, because then the specification is gone as soon as you pretty much turn those web pages off. But what happens here is that we can gather information whether or not the instrument or the core facility is in existence, because a lot of these things happen automatically. So once the PID is identified, once that metadata is there, it can be used to either text mine, or it can be used to pull information in other ways. The authors often use the RIDs for these things. They are directed to do so by their by their journals. And sometimes they use names, which we also have as a piece of the metadata. So we can actually train some classifiers, some AI classifiers, in order to try and find those pieces of information from articles. And then there are various identifiers that we have, which can also be used to pull these things into a list. So next slide, please. And just to give you a flavor for what kinds of things and what kinds of ways we can actually pull papers by, if the author uses an RRID, whether or not the paper is published in an open access journal or in a closed access journal, about 2 to 3 students from my team will typically be able to find that. They have a little bot that they can run on top of any paper that they have access to. And that pulls in all of those citations. But of course, they are not going to search for every alternative name. They're not going to search for all of these crazy things. What they're going to search for is the RID. If the author used name of a particular entry, then we actually do have in the open access subset of PubMed Central, we actually have an automated pipeline that runs monthly that pulls those information in. And if it passes some very basic validation checks, those things are then pulled across into each of the records. Alternative identifiers are a little bit easier. So there are DOIs and there are many other types of identifiers, including core marketplace identifiers for the various facilities and various other things. Those are all brought in, again, monthly. Authors may also use URLs. We tend to have, for the older cores, we actually might have 5, 6, 7 different URLs that maybe are no longer useful, but they have been the URL in the past. I think, Amanda already mentioned link rot, and we have actually quantified link rot. It turns out that within 2 years, 25% of your links are broken within 2 years of publication. So that means that, a lot of these old links are, they're no good anymore for actually getting you back to that facility, but they are very good if you have them listed along with a record, because somebody may have actually used that link in a publication, and that is how they're identifying your core. Next slide, please. So before everybody gets too excited, what I would just like to highlight here is that there are robots that we have that can actually go very deep into these papers, and they can find a lot of great things from our metadata. I'm sorry, there appear to be some blue angels above me. This is the negative part of living in San Diego. I apologize. But what happens is within the open access subset of PubMed Central, we have a lot of room to be able to find information about those entities. But if something is not in that PubMed Central open access subset, then we are much more limited. And then there's our little icon for our great students. Our students are wonderful, but they really just search for the RID itself. So I wanted to take the next 2 or 3 slides. So next slide, please. And just ask the question, why would anybody care about this thing, right? Okay, so you can have a page, you can have it on Core Marketplace, you can have a ROR record, you can have an RRID, but why do you care about this? So if you go to the next slide, it turns out that when we've looked, and then cores would be here under tools, when we looked, and this is just, you know, now a very old figure from a very old paper when we first started the RRID initiative. And if you can just focus on that antibody bar right there, the percent identifiable. So this means that a person would be able to go into that paper and identify that particular resource or tool. And what we find in the first, and this is the first 100 papers that were ever published with RRIDs, the identifiability between the issue of the journal before putting in the RRID as a request or requirement versus those that are requiring changes from about 50% identifiability to 90% identifiability. It's not quite as good for tools, but it is kind of amazing. Next slide, please. And so we actually quantified this a few years after to take a look at what the impact may have been across the totality of the literature. And so what you see here is not identifiability in the same sense. This is identifiability of a reagent by catalog number specifically. And so we knew that in the early 2000s, the percentage of catalog numbers in that scientific literature was very low. Here we quantified it, it was between about 10 and 15%. So 10 to 15% of reagents of antibodies would be actually listed that with having a catalog number. And then that very slowly changed over time. And in 2014, when we started the RRID initiative, we had, you know, somewhere in the order of about 20% of the antibodies that were able to be associated automatically using with a catalog number. And again, this is the totality of the open access literature up to this point, which is 2019, and this is about 300,000 records. And what you can see here with these, you know, other kind of more fuzzy lines, these are the individual journals. And you can, one thing that you can, I think, pretty clearly see is that these 2 green lines, and this is Cell, the journal Cell and the journal eLife, which both came up with a pretty stringent requirement for RRIDs. Now that drove that percentage of antibodies that were listed with the catalog number from this kind of an average rate of somewhere in the 20 percentile up to the 90 percentile. And what that does is it actually improves and pulls on the rest of the literature. It starts to actually turn how investigators think about what kind of information that they're putting into the papers. It drives more persistent identifiers. Now, these are not all RIDs, and I'll show you what happened to mice in the same at the same time period. So if you can go to the next slide. So what happens here is now we're looking at specifically the Mutant Mouse Regional Resource Center mice. And this is a study that we did manually looking at 2,400 papers. This is every paper that we could find in PubMed Central that had used one of these mice. And we categorized them into 4 different categories. Each and every single one of these mice was categorized into whether it was a nickname, where we could tell, and you could see that here on the right-hand side, the orange nickname, this is a nickname for this mouse. We can see that it's MMRC, but we actually cannot get back to an individual organism when the authors use this nickname. And then here's the full name, and if they use the full name, they're quite long, as I'm sure many of you know. If they used it, typically we were able to find the actual RID associated with this mouse. Catalog numbers tend to be easier, also very much easier to find, but they're not standardized, so this is not machine readable. And then the RRID tended to be here, and you could see that this actually is machine readable, so this is very easy to actually be able to detect. And one thing that you want to maybe jumps out at you already is that in 2011, half of the mice were not identifiable uniquely. So half of the papers using mice from this repository were not reproducible because of this reason. And then what we've done here with 2014, there were only a couple of papers with an RID. But what you can see is that percentage with RIDs is growing, and it's growing leaps and bounds, roughly. We're just over 50% in 2024. But the other thing that's growing is it's not actually that RID citation is not cannibalizing the catalog number, it's actually cannibalizing the nickname. So the nicknames are way down to about 5%. And this is amazing, because now 95% of the scientific literature using these reagents have it in their, those authors have it in their head that they're going to do a better job identifying the things that they're using, which is, I think, quite remarkable. And this is why we care about this stuff. The other thing that happens, next slide, please, is...
Anita, sorry, we are 7 minutes over, so can you...
Oh, goodness gracious. Okay, I am not going to talk about reagent warnings, but thank you so much, and I am so sorry I went over. This is my goal.
We're passionate about it, for sure.
My goal was definitely not to do that, so I apologize, and I will just thank all the members of my lab who will all tell you to use RIDs.
Okay, I'm sorry about having to abruptly interrupt you. So obviously we are not going to have time for questions right now, because we're over time. But I actually was I'm chatting with Judith about the possibility, and I will send an email to Nate, Anita, and Amanda on the possibility of having a follow-up session. We have something called a drop-in session, in which actually is just purely for people to just ask questions. It's more like very, very informal. There are no presentation. I mean, obviously, if you need to show a slide, you can, but it's about really people asking questions. And we have heard from the community that people still have questions and sometimes doubts about how to use RRIDs for facilities and associate instruments. So I think it would be really, really well received. So I will follow up with an email if you guys are interested. I'm hoping that we can announce it soon as a way of, people will be able to see the recording of this and then actually have questions. All right. Thank you so much. Thank you for participating. Thank you for presenting. This was really extremely useful, I think, and we need to do more of this. Okay.
Yes.
Bye.
Thank you so much, everybody. That was amazing. Really amazing.
Thanks for having us.
Thank you.